Hello dear students, this is Dr. Adwet and welcome to the second lecture of Cell, the Unit of Life. So, without wasting any time, let us first talk about all the things that we discussed in the previous lecture. The first thing we saw was English as a language of the peasant, as a language of the common people, has undergone numerous transitions in the last 1000 years. We went from Old English to early modern English to about late modern English, which is what we are using today. And as English was transitioning, it was getting a lot of influences from the Latin language, which was the language of the scholars or the language of the people who were learned, or the official language of the Roman Catholic Church. So Latinized words or scientific words which were in Latin at that time started entering into the English vocabulary. And this is the reason why we see a lot of Latin words in our study of biology. In fact, biology comes from the two Latin words bios, which means life, and logos, which means study of. Then we saw that all living organisms need some sort of energy to survive. The form of energy that most of the living organisms are going to use is a chemical form of energy called as ATP, adenosine triphosphate. The universal reaction to make ATP is the first reaction shown above here. That reaction states that you first require glucose, which you will oxidize using oxygen to give you carbon dioxide, water and ATP. So if you need ATP, you need the starting reactant, which is glucose. All living organisms will do this reaction. 99% of living organisms will do this reaction called as respiration, but that requires glucose, and the process by which glucose is made is not done by all living organisms. So now look at the second reaction by which glucose is made below respiration. The second reaction states that you take carbon dioxide, combine it with water in the presence of sunlight and chlorophyll to make glucose and oxygen. So the first reaction, respiration, all living things do, but the second reaction, which makes glucose, without which you cannot do respiration, is not done by all living organisms. That second reaction is called photosynthesis, and it is only done in the presence of sunlight by plants which contain chlorophyll, the green pigment chlorophyll. So all living things do respiration, but all living things do not do photosynthesis. Based on this, life is broadly classified into two main groups. Those, as you can see on the screen right now, which produce their own food, and those which do not produce their own food. Those which produce their own food means that they will first make glucose, and then they will use that glucose, use respiration to make ATP. Those which are not going to produce glucose or produce their own food will still do respiration, but they will not do photosynthesis. So this brings us to the next three Latin words, which are auto, hetero and trophos. Auto means self, hetero means different, and trophos means to eat. From this we get the first new word which we saw last time, which was autotroph. Autotrophs are those organisms which will eat that food which they have prepared themselves, to eat what is prepared by self. Because they make their own food, they make their own glucose, they do not need to go from one place to another in search of glucose. So they don't need to move about from one place to another. Such living organisms are called as plants. On the other hand, you are going to have those organisms which do not make their own food but depend upon different other organisms to provide them with glucose. These which are going to eat food prepared by others are called as heterotrophs. Heterotrophs will compulsorily have to move from one place to another because they cannot make food on their own. These are going to be called as animals. The Latin word for plant is botan. The Latin word for animals is zoon, and that is how you have the study of plants called as botany and the study of animals called as zoology. So biology, study of living organisms, can be broadly classified into botany and zoology, study of plants and study of animals. Next, we went on to have a detailed discussion about cell, and we saw that cell is the structural and functional unit of life. I explained to you that cell can simply be called as the unit of life, and we saw that unit has three meanings. The third one is going to be the one which fits in our definition, and that meaning was the smallest particle showing property. And that is how we came to the definition of a cell. Cell is the structural and functional smallest particle showing property of life. Is cell the smallest thing inside a living organism? No. But is cell the smallest living thing inside an organism? Yes. So I hope that this distinction is now clear with everybody. So this finishes everything that we have done in the previous lecture, and today's lecture starts now. The first thing that we have to do today are a few more basic terms in Latin, and they are micro, macro, mega and magnum. Very small, small, big and very big. Micro, macro, mega, magnum. Very small, small, big, very big. So easy enough to remember, and you've heard these words before. So now if you have just a clear understanding of this, micro is very small, macro is small, mega is big and magnum is very big. The next word is a Latin word which also you've heard before, called as scopion. The word scopion means to see. So I think you know where I'm going with this. There's a word for an instrument called as a microscope. Microscope again does not mean to see very small. It means an instrument which is used to see very small things. So why are we doing this right now? Because you know that cells are so small that they cannot be seen with the naked eye. Cells are a microscopic structure. So cell, which is the structural and functional unit of life, is a microscopic structure. So that takes us today into a journey talking about the history of microscopy. So microscopy, or study of microscopes, or study of microscopic things, is going to begin with glass. Do any of you know when glass was invented? How long back? 1000, 2000, 3000 years ago? It is believed that glass basically was invented around 4000 years ago. Somewhere in Arabia, it is estimated that glass originated, or glass was discovered from, silica, or how glass could be made from sand. That was discovered somewhere around 4000 years ago. Clear glass, glass without impurities, or glass of very fine quality, glass which has good transparency, has been in existence at least for the past 2000 years. And in the very beginning, 4000, 2000 years ago, till about this time, in fact till about 700, 800 years ago, even though people knew about the optical qualities of glass, it was not used for any scientific research. It was usually used for making objects like cutlery, basically bowls and vase, or cups, things like that, or it was used in jewelry. Glass was not used for its optical qualities, at least not mainstream. Somewhere in the 1300s, early 1300s, people realized that glass has optical qualities with which you can magnify things, or with which you can bend light. Once people started knowing about that, as a person ages, the eyesight becomes weaker and weaker. So people realize that these optical qualities can be useful to improve the eyesight, or maybe it may help to magnify a few things. So primitive lenses were used in making eyeglasses, or were used for eyeglass production around as early as 1299. So in the later part of the 13th century and in the early part of 14th century, people already were using eyeglasses. But it was not like how we have today, that you go to a optician and they will give you your prescription of glasses with which you can see clearly. No. So in the early 14th century, glasses or glasses for improving vision, optical glasses, were used, but they were so primitive and the method was so crude that you went to an optician, he would have multiple glasses, ऐसे ही पड़े हुए, not knowing कि क्या diopter है, क्या power है, क्या nothing of that sort. You just go to that person and you try, and then you see 3 or 4 or 5 or 6 or all of them, and अगर तुमको कोई नसीब से fit बैठ गया तो you buy it. This is what was happening in the earlier 14th century, sometime in the year 1320, 1330s, at around that time. This is what was happening. Of course, as time went by, this technology would improve because people started grinding glass. Now, what do you mean by grinding glass? Taking something and taking the glass piece of your choice and then scrubbing it against that so that you can mold the surface of the glass. So you are grinding the glass to give it a shape that you want. So once people figured out that if you grind the glass, you can improve its optical qualities, slowly that process began to improve. So that brings us to this man. And now you're going to see a lot of black and white, awesome images of cool dudes from the past. And this guy was called Hans Lippershey. He was a Dutch optician, widely credited as the first person to patent a telescope in 1608. So this guy is credited with the discovery of the telescope. The telescope that he made looked somewhat like this, and so in the earlier 17th century, sometime in the 1610, 1620, Dutch lens makers fashioned a telescope by pairing a concave and a convex lens in a tube, as you can see above. So inside here there were two lenses, there was a lens here and there was a lens here. So one was concave and one was convex, and although the instrument was not much more than a crude spyglass having a magnification of only 7 or 8 times, it was still a huge breakthrough at that time. Now, I want you to understand this simple funda here, that in the early 14th century, so this all that we are talking about, whatever is being talked, is 17th century. So since around 1310, 1320s, people already knew that glass had optical property and they were using them as optical glass to help improve their vision. So single lenses were already being used. From the 1320s to the 1420s to the 1520s to now the early 1600s. So we are talking about 300 years. So of course during this time people were playing with multiple lenses. Crude spyglasses like this were also made before, but this guy Hans Lippershey was the first one who kind of told an official government office that this is something that I have made. So he was the first one who went to the government office to register this device, and that is why we have a record that this man was the first one to invent the telescope. So I'm guessing that there were people before him, but they are not given credit because his is the first recorded instance of somebody using the word telescope and getting their design to the government saying that I am the inventor of this. So that's Hans Lippershey. Then came this guy, Zacharias Janssen. Now Zacharias Janssen was also a Dutch optician, and he is credited with the invention of the compound microscope in the 1590s. So if you know a little bit of optics, you would know that if you were to take a telescope and if you were to simply invert it, it will have very slight microscopy abilities. So the idea is that if you use a telescope like this, it is going to make something which is far away appear bigger, and if you were to just reverse the optical design, it will magnify something which is quite close. So like I said, even before Lippershey, there were going to be people who were playing around with this. So people knew about telescope and microscope before these two guys, Lippershey and Janssen. They knew about these properties, but these were the first guys who were credited with it because we have a written record somewhere in the government diaries about these people. So he is widely credited to be the inventor of the compound microscope, and this is what his compound microscope looked like. So it had two lenses, a lens here and a lens here. this one which was close to the object, that lens was called as the objective, and the lens close to the eye was called as the eyepiece. And in between these two, you can see that there is this thing called as a body tube. So if you were to see here, this tube can move within this body tube, and this tube here can also move within the body tube. So the two ends could move front and back, and the body tube would remain fixed. So you can move this part, the eyepiece, and this part, the objective, individually. So this allowed you to adjust the focus and view something small in a better manner. So this was the compound microscope, and this was already discovered or already formulated, invented around 400 years back. That was in the 1590s by this person called as Zacharias Janssen. Okay, that brings us to the next scientist, who is this person you see on the screen right now. He is Robert Hooke. Robert Hooke was living around 1635 to 1703, so in the later part of the 17th century. Robert Hooke was based in London, and he was one of the scientists at the Royal Society. Now the Royal Society is one of the most prestigious and oldest scientific institutes or educational institutes in Europe, and it was founded in 1660, and he was one of their earliest scientists. He was kind of a polymath. A polymath is basically a person who dabbles in a lot of sciences. So he was deeply interested in natural history. He, in his younger days, or when he was a young boy, he had also worked at the local artist. So he was trained as an artist. He was an architect, so he was deeply passionate about architecture, and at the same time he was also interested in a lot of natural sciences. So he was one of the first scientists working in this premier institute in Europe at that time called as the Royal Society. So Robert Hooke wrote this book and illustrated it in 1665 called as Micrographia. Now, from the little bit of history that I've already told you, microscopes were in existence from the 1550s. The official credit goes to the person which we just saw, Janssen, which was somewhere in the 1590s. And from the 1590s to the 1650s, the design in microscopes had gone on improving. In fact, this was Robert Hooke's personal microscope. So as you can see, it's just a modified version of that tube which you saw, and even this is going to have one lens here called as the objective and one lens here called as the eyepiece. So Robert Hooke in 1665 took a lot of ordinary everyday items, put them here, and since he was trained as an artist, he illustrated them beautifully in this book called as Micrographia. Remember that this is 1665. It has just been about a couple of hundred years since the discovery of the printing press, and this was one of the first books with illustrations of those microscopic worlds which nobody had seen before. So this book, if I was to show you a few pages from this, if you were to open it up, this is how the illustrations were placed inside the book. So you can see the book is quite small, and like how we have today, this has a few folding pages inside it. So if you were to open the book and if you were to fold it out, you would see this picture of a flea, and this is how it looks. So people at that time knew that what was the size of a flea, but because they had no ways of magnifying it, they had no idea about its intricate beauty. So when Robert Hooke actually put the flea under the microscope and magnified it to this size and drew this illustration by hand, the entire public went berserk because nobody had seen anything like this at all. There were no instruments of any kind before this which could show this to the world. So the first time that the world was actually exposed to microscopic structures. In fact, this was not the only picture in the book. There were many beautiful illustrations, like this picture of an insect. The next picture is that of a louse which lives in human hair. So what you see it holding here is an actual human hair. So when Robert Hooke actually drew all this, illustrated it, there were other beautiful pictures like this picture of the head of a fly. So when he did all this, all right, it literally took the public by storm. So this Micrographia was an extremely popular book at its time, and even though it was very expensive, a lot of people bought it because there was simply nothing like it before. In fact, there has been nothing like it even after. So this book was extraordinary. The first, in fact, the very first image that you see in the book is this. So this is a pencil tip. This is a full stop which was made by an ink pen which you saw under the microscope, and this is a knife's or a razor's edge. So these everyday objects he was able to illustrate, see under his microscope, and literally, like I said, take the whole world by storm at that time. Now in his book Micrographia, he also drew a picture, or he also showed the instruments that he was using. So this is his instrument that he had used, the picture which I had shown you before. So if I was to magnify this, this is what it looked like. So now the idea here is that there is a glass piece here, there is a glass piece here, so there is an objective here and there is an eyepiece here, that Now the other thing is that here is a globe of glass, or basically a globe which is filled with water, and here is a candle. So what happens is that the light from the candle through this gets focused at this point. So the light from the candle passes through the globe, which focuses the light at this point where he is putting his specimen. Then the light from here reflects into his microscope and he is able to see whatever has been placed, the specimen. Now this point is important because what this means is that whatever Robert Hooke was seeing was via reflected light. Reflected light. Light is coming, hitting his subject, and then reflecting off it, passing through his microscope, and that is what he is seeing. So this was Robert Hooke's book called as Micrographia. Now, why am I discussing all of this in so much detail? The reason is that in this book, on page number 112, there is something very important. So this is page number 112. This is a expansion of an illustration that is coming from page 112, which you can see here, and I'll tell you more about it in a minute. So this is page 112, this is page 113, 112 and 113, and in between these two pages was this picture here. So what is this picture here? This picture is an extremely thin slice of the outside part of a tree which is called as cork. So what he has done is he's taken a razor and he's taken the outside part of a tree and cut a very thin slice of it and placed it here. In fact, I would like to read out what he has written about this. So I'm just taking this paragraph from page 112 and magnifying it here. I took a good clear piece of cork and with a pen knife sharpened as keen as a razor, I cut a piece of it off and thereby left the surface of it exceedingly smooth. Then examining it very diligently with a microscope, methought I could perceive it to appear a little porous, but I could not so plainly distinguish them so as to be sure that they were pores, much less what figure they were of, blah 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 blah. Now as you can see, this is the 1650s, 1665 when this book was written, and you can see how poetic the language is. So this is what I was talking about in the previous lecture, Old English to early modern English. So this is an example of early modern English, and that is why you see things like methought, etc. The grammar is very weird. Okay, so this is what he wrote about them. Now the important thing here is that on page 113, if I was to magnify this section here, this part says next in that these pores or cells, in these pores or cells, this is the first time in recorded history that the word cell has been used. Robert Hooke was the first person who used the term cell. It is believed that he used the word cell because he was an architect. So when he saw this in his microscope, when he was seeing this through his microscope, this circle, he thought of these things which he saw as these tiny compartments where monks live. So you remember he was an architect. So basically, if we were to look at this architectural drawing of a monastery, all these little compartments that you see here are rooms where the students who are wanting to become monks were going to stay. These things, by the way, were called cella. These quarters or these rooms where these student monks used to live were called as cella. And that is why it is widely believed that he gave the term cell to these structures. Now what he saw here were actually the dead sclerenchymatous cells of this plant. So we will understand what these words are in the future, but he saw these dead plant cells, and because of these dead plant cells which he saw containing nothing, and it appeared to be like empty rooms to him, he gave it the term cell. As you can understand, this is a misnomer. A misnomer is a name which has been wrongly given. So of course ye naam galti se diya hai, because if he is calling them as cell, and from this we call all such structures as cells. Cells are not empty, they are of course going to be filled with something, they are not empty rooms. But since they looked like empty rooms to him, he gave the term cell. So Robert Hooke was the first person to use the term cell. And that is why I wanted to give you this little history which explains to you clearly that Robert Hooke is the first person who used the term cell in his widely acclaimed book in 1665, which was called as Micrographia. Alright, moving on. The next person is this man called as Anton van Leeuwenhoek. And Anton van Leeuwenhoek was living at the same time as Robert Hooke, around the same time in the 17th century, between 1632 and 1723. Now the first interesting thing about this man is that he was not a trained scientist of any sort. In fact, he was not even a scientist. He was a Dutch fabric merchant in this city in Netherlands, Holland, called as Delft. Now, as a merchant for clothes or as a draper, one of his most important jobs was to examine the cloth with a magnifying glass to pick out any defect in the cloth. Because if you were going to use the cloth for royalty, it should be without any defect. So it had to be very carefully examined, and for this purpose he was of course using magnifying glasses. So I just want you to know that this man was not a scientist, he was a draper, and he was well oriented with using magnifying glasses. This point is going to become important in a few minutes. Okay, so again, before talking more deeply about him, Robert Hooke was living in London between 1635 and 1703. He was a polymath, he was a scientist with the Royal Society, he was also an architect, he was interested in microbiology, he had a lot of interests. And then we have this guy who is Anton van Leeuwenhoek, who in fact is more widely remembered than him, and you will understand why. Was living around the same time from 1632 to 1723, and he was based in Delft. So if you were to see the map of this region, this is the United Kingdom or Great Britain, England, this is London, and this here is the Netherlands, and they are separated by the English Channel and the North Sea. And here, somewhere here, is where Anton van Leeuwenhoek was living, and this London is where we have Robert Hooke. So I hope you understand where these two people were at that particular time in history. Now let's continue talking about Anton van Leeuwenhoek. So this is the microscope which Robert Hooke was using. It had two lenses and it was trying to magnify the image by using reflected light. This had a magnification power of about 20 to 30x. Matlab it could magnify the image, whatever you are seeing through it, about 20 to 30 times. Now Robert Hooke was using this, but Anton van Leeuwenhoek was going to use single lenses, single convex lenses, and they, as you rightly know, are called magnifying glasses. With this single lenses, Leeuwenhoek was able to magnify up to 266 times. Let that sink in. 2 lenses, little complicated design, able to magnify only up to 30 times. And here we have another person who is not a scientist, who started using only 1 lens but was able to achieve magnification of up to 266 times. So now how is that possible? So I'm going to give you a quick lesson in optics in the next slide. So what you see on your screen right now is a convex lens, and this little video is going to show you how a convex lens is actually going to magnify an object. You already know this, that if you take a convex lens and you place something in front of it, because of the optical properties, it will magnify that image in front of your eyes on this side of the convex lens. So if you were to see a ray diagram of that, which you've seen in school, you place an object in front of it, you get a virtual image of that object, which is produced as follows. So I hope everybody is clear with this. Now, the important thing is, if you were to increase the curvature of the lens because of its optical properties, the size of the magnified virtual image will go on increasing. I repeat once again, if you increase the curvature of the lens, the magnification of the image increases. So have a look at the animation and you will understand this clearly. So as you can now see, the curvature is going on increasing. Can you see that? So as you increase the curvature, the size of the object goes on increasing and increasing and increasing. So how do you increase the curvature the most? The simple answer to that is you simply change the lens to a sphere. So if you have a convex lens like this, and if you go on increasing the curvature, at a point it is going to become a sphere, and the sphere will have maximum curvature. Can you increase the curvature even more? Can you make the curvature even more extreme? And the answer to that is you make the sphere smaller and smaller and smaller. So as you make a convex lens more curvaceous, it will magnify images even more. So a sphere is really going to magnify the image, and if you make the sphere smaller, the curvature will become more and more extreme, which will result in a much higher magnification ratio. So you can continue seeing that in the animation now. So as you decrease the size, the curvature becomes more extreme. So I hope that this is now clear with everybody, and this is what Leeuwenhoek was doing, using extremely tiny lenses which were perfect spheres, because he had understood how to grind them properly. Nobody knows till date how Leeuwenhoek was grinding his lenses. There is no record of that, and he has kept that a secret, but we know that he was doing this because in the lenses or in his little microscopes which survive today, we know that the lenses were highly polished, but we don't know how he did it at that time in the 1650s. So this here, what you notice in this image now, is his microscope, and this is that tiny little lens we are talking about. This whole thing which you see here is only about 2 inches, and this is only about 2 to 3 millimeters in diameter, this little lens which you see here. Let me just play this video to show you a magnification of this. So this is his lens, the extremely spherical lens that I am talking about, which is extremely small. And as you can see, in front of that, there is this small area here on top of which he could put whatever he wished to see or magnify. So he could literally take a drop of water and put that drop of water here and see what is inside that drop of water. Now there was only one problem with this, and the problem was that if you had an instrument like this, okay, first let me show you how big this instrument was. So this is the instrument, and this was the design of the instrument. So like I said, this is only about 2 inches, and this is only about 2 to 3 millimeters. So what he took was, he took these two metal plates, and in between that you can see that he has sandwiched this. He has sandwiched his lens, the spherical lens with very high curvature, and here is where he was going to put his specimen. So this was the design of his microscope. How big was this microscope? It was just this big, like literally this big, this big. And the problem with this microscope is, if you wanted to see anything, you would have to place it extremely close to your eye. In fact, you needed to almost touch that lens, that spherical lens, to your eyeball. You had to bring it that close. So using this was going to be extremely, extremely cumbersome and deeply uncomfortable. But Leeuwenhoek made about 300 to 500 of these microscopes in his lifetime. So we know that because he had written a few letters which you will come to know about. So we know that he had about 500 of these. Very few of them survive, and that is why we know that he was using such kind of microscopes. So this is how close it had to be to the eye. And we also have a picture of him with his wife showing how he was using this microscope. So he was also looking through the microscope and he was drawing pictures. Now, I want you to remember that when he was doing this, Micrographia had already been released. So we are talking about 1665 having already gone. This is sometime in the early 1670s, around 1672 to 1675. So during this time, he was doing all of this research on his own, out of his own curiosity. Now, one day, it has been, written in a few books, a few anecdotes, that one day he literally took some plaque out of his teeth and he observed it under this microscope. So he had seen a lot of other things. He had seen pond water, he had seen stuff like semen, animal semen, he had seen ox tongue. So he had seen a lot of things. But one day he literally removed some plaque from his teeth and he placed it on the pin and he observed it. And what he saw completely changed entire understanding of our world. What he saw was little cells like these, which were probably bacteria. And he saw that they were moving about through his microscope. He saw that they were moving about. He gave them the name animalcules. And he called them as animalcules because, as you can see, they clearly looked like little animals. And that is why he called them as animalcules. He was observing all of this somewhere in the 1670s, around 1672-73 के आसपास. He wrote a 17.5-page letter to the Royal Society about his observations. And he sent it to the Royal Society, where Robert Hooke actually read all of it. And Robert Hooke was amazed and did not believe this, that there was a man sitting across the English Channel who was not even a scientist, who is claiming to see these little animalcules, living things in his, plaque. So Robert Hooke sent a few learned men or men with good reputation, कि please go and check out whether what this man is saying is true. So Anton van Leeuwenhoek had to actually show all these people who came from Europe, from London, कि Baba, I am not lying, you can also have a look at it. And when they saw it, they were equally amazed, because this was the first time somebody had seen a living cell. Robert Hooke had seen dead cells, but this was the first time somebody actually saw a living cell. And when all these people reported back to Robert Hooke, the finding of Anton van Leeuwenhoek were published in the Journal of the Royal Society. In fact, in 1780, he was also granted membership into the Royal Society, even though he never went to London. But he was also considered as a scientist by the Royal Society. So the reason why I find his story extremely fascinating is because this is an ordinary draper who was just motivated by his curiosity to learn about the world, and I find that extremely interesting. So Anton van Leeuwenhoek, as of today, has more recall and people remember him more as compared to Robert Hooke, who was an actual certified scientist, simply because he was the first one to see the living cell. And that is why his name goes down in history, just because he was so curious. So that brings us to this slide, and before I show you the content of this slide, I hope everybody noticed that in that corner there, you can see this symbol appear. Now, this symbol, whenever it will show up on a slide in my lecture, means that the entire content on that slide is from the NCERT textbook. So this becomes very important, because whatever is here can be asked to you in your NEET examination. So this is directly from the NCERT textbook. So the NCERT textbook says, Anton van Leeuwenhoek first saw and described a live cell. Now the reason why this is important, and whatever we've done so far about the history of microscopy, is just so that you understand this bit here. So Anton van Leeuwenhoek first saw and described a live cell, but he did not give the term cell, he did not coin the term cell. The term cell was coined by Robert Hooke, the earlier scientist that we saw. So keep that in mind. So Robert Hooke can also be asked to you in NEET, even though it is not written in NCERT, because he is widely regarded as the person who first saw the cell or gave the term cell. But this person, Anton van Leeuwenhoek, was the first person to see living cells, and that is why his name is more pronounced in history. Then in 1833, you had Robert Brown, who discovered the nucleus. So NCERT straight away jumps, like a full century, 70-80 years to 1833, where Robert Brown discovered the nucleus, and the invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. So this little history lesson was important for you, so that you understand how our whole understanding of cells began. So now, a quick recap of the words we've done so far. We saw bios is life, logos is study of, auto is self, hetero is different, trophos is to eat, botan is plant, zoon is animal. Today we saw micro is very small, macro is small, mega is big, magnum is very big, scopion is to see. And now a new word which you had not heard before, cyton, which also means cell. So this brings us to our next definition, which is cytology. So what do you think is cytology now? Cyton is cell, logos is study of, so cytology is study of cell. But study of cell would not be possible without a microscope. So it is not just study of cell, it is microscopic study of cell. It deals with the study of detailed cell structure, cell organelles, their function, cell division, etc. So now you know what is cell, and you also know what is cytology. This brings us to our next topic for today, which is the cell theory. Now, what is this thing called as the cell theory? So, again, two more dudes in black and white looking awesome. The first one is Matthias Schleiden, and the second one is Theodor Schwann. Now, both of them were living somewhere in the 19th century. So Schleiden was between 1804 and 1881, whereas Schwann was between 1810 and 1882. So living around the same time. And NCERT textbook says that Schleiden is a German botanist, whereas Theodor Schwann was a British zoologist. Now in reality, I did a lot of research on this. Schwann was also German. So I don't know why they've written British here, but we will go along with it because that is what is written in the NCERT. So Schleiden, German botanist, Schwann, British zoologist. One was studying plants, the other was studying animals. Remember, all of this is happening somewhere in the mid 1800s, 1830s के आसपास ये लोग अपनी लाइफ में रिसर्च वगैरह कर रहे होंगे. So 1830s, please remember, is 200 years from, or 250 years from 1590, when Janssen discovered the microscope. So by the time these people were working as scientists, it had been around 250 years since the microscope had been invented and had been improved upon. So by this time, they had access to good quality, decent quality microscopes with which they were working. So of course, since Schleiden was going to be a botanist, he would be seeing a lot of plant tissue under the microscope, and since Schwann was basically working mostly with animals, he was going to see a lot of animal tissue in the microscope. So in 1838, Schleiden examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. So Schleiden in 1838 observed that majority of the plants, in fact everything that he saw under the microscope, was made up of cells. Then came Schwann, the British zoologist, who in 1839 studied different kind of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. So basically, he independently, a year later, so Schleiden was 1830 and Schwann is 1839. So a year later in 1839, he independently came to the conclusion that animal cells, or rather animals, were also made up of animal cells. He in fact, under his microscope, also noticed that they have a outer thin membrane called as the plasma membrane, which we today know as the plasma membrane. So again, an important point about Schwann, he was the first person to describe the plasma membrane. So Schleiden ने बोला कि plants are made up of cells, Schwann ने बोला कि animals are made up of cells. In fact, when he came to know about this, that all animals are going to be made up of cells, he wrote letters to all the other scientists which were working in the same field. And when he wrote letters to everybody in the scientific community at that time working in that field, he got a reply from Schleiden. And that is how the two came to know about each other, and that is how the two of them started working together. So he also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. So this is after his association with Schleiden. So he is the first person to say that animal cells में मुझे plasma membrane दिखा. He is also the first person to say कि plant cell में बाहर मुझे cell wall दिखा. These things we are going to do in more detail in the next lecture. But today I just want you to remember that Schwann was the person who first described the plasma membrane and also the cell wall in the case of a plant cell. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. So this was Schwann's contribution in 1839. So both these scientists, Matthias Schleiden, who was a German botanist, and Theodor Schwann, who was a British zoologist, both of them together formulated the cell theory. So what is the most important point of the cell theory, as you probably have understood by now? All living organisms are composed of cells or products of cells. All living organisms are composed of cells or products of cells. This was the most important point of their theory. However, they could not explain how new cells were formed. So they were able to say that everything was going to be made up of cells, whether it was plant or animals. They were not able to explain, or they did not know that where were cells coming from. And that brings us to the next scientist, whose name is Rudolf Virchow. Now, Rudolf Virchow in 1859, mind you, which is a good 16 years after these people were, talking about all living things are made up of cells, he said that cells divided and new cells are formed from pre-existing cells. In fact, in his account, he also wrote omnis cellula e cellula. So this is a Latin line which all of you are supposed to remember. Omnis cellula e cellula. New cells come from old cells. So the literal translation is new cells from cells. So omnis cellula e cellula means new cells from cells. So this was his research, where he concluded that cells divided and new cells came from pre-existing cells. So he modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. So what is the cell theory? Number one, all living organisms are composed of cells or products of cells. And number two, all cells arise from pre-existing cells. So these are the two main points of the cell theory. So let's have a quick recap of what we've done today. We saw what is biology, what is botany, what is zoology, and what is cell in the first lecture. Today we saw the history of microscopy, we saw what is cytology, we saw what is the cell theory. This, dear students, concludes today's lecture. I hope you've understood everything that we've done in today's lecture, and I cannot wait to see your smiling faces in the next lecture. Thank you and God bless.